Hey everyone, I wanted to provide a quick little video here to serve as an add-on to my other Chapter 22 videos covering the respiratory system. In recent semesters, I have added a section here on the dynamics of hemoglobin and internal versus external respiration, and those were not covered in my previous videos, so I wanted to provide a quick little add-on here just to kind of supplement your education here. So let's go ahead and get into this, understanding that at this point you've probably watched all the other videos in this chapter. So dating back to earlier chapters, we have talked about hemoglobin before. So we know that hemoglobin is the major oxygen-carrying molecule that is found inside of red blood cells, and it serves as the major carrier for oxygen in our blood. So we also understand that hemoglobin goes through periods in which it binds to oxygen to form oxyhemoglobin, but it also goes through a period where it releases oxygen to become deoxyhemoglobin. The idea that hemoglobin can both bind to oxygen with high affinity and then also let go of it when it needs to for the sake of delivering oxygen to needy tissues is actually a very important quality that hemoglobin has. The graph that you see here is known as a dissociation curve. On the y-axis, you can see the relative saturation of hemoglobin with oxygen, with the bottom of the y-axis 0, indicating that no hemoglobin is carrying any oxygen at all, and the top of the y-axis at 100%, indicating that every single hemoglobin is fully loaded up with 4 oxygen molecules simultaneously. On the x-axis, we're looking at the partial pressure of oxygen in the blood. We've seen before that in the arteries with what we consider oxygen-rich blood, the partial pressure should be about 100 millimeters mercury, whereas in the venous blood with what we would consider to be deoxygenated blood, this is going to be closer to 40 millimeters mercury. So the idea here is that there is a sigmoidal mathematical relationship between the partial pressure of oxygen and the oxygen saturation of hemoglobin. What that basically says is that at very high concentrations of oxygen in the blood, such as in the arteries and in the pulmonary capillaries, hemoglobin is going to have a very, very, very high affinity for oxygen. So when the partial pressure of oxygen is very high, hemoglobin will be fully loaded up with oxygen. But at lower concentrations of oxygen, such as in the veins and in the tissues, where concentration of oxygen is much lower, the affinity that hemoglobin has for oxygen is much, much, much less. So this is going to be an important dynamic for us to consider because we are going to be talking about a number of factors that influence hemoglobin's affinity for oxygen and how these factors at different points in the body really set things up perfectly so that hemoglobin can bind oxygen for when it needs to and let go of it when it needs to as well. So an important thing to understand as it pertains to this dissociation curve is that anything that would cause the curve to kind of shift towards the left, that means that hemoglobin has a higher affinity for oxygen. And anything that shifts the curve to the right would cause hemoglobin to have a lower affinity for oxygen. So the closer this green curve here is towards the left side of the graph, the higher the affinity is, and the closer it is to the right, the lower the affinity is. Okay, so first let's talk about the difference between myoglobin and hemoglobin. Myoglobin is a molecule that we discussed briefly back in Chapter 10 when we talked about muscle physiology. So myoglobin is a single subunit polypeptide that also serves as an oxygen-carrying molecule in slow-twitch muscle fibers, and you'll recall that it's responsible for their red color and their capacity to not fatigue very easily. So myoglobin is very, very similar to some of the hemoglobin subunits in the sense that its polypeptide structure and folding is very similar, and it also contains a single heme prosthetic group that allows it to bind to a single molecule of oxygen. The major difference, though, is that hemoglobin is a multi-subunit protein in which it consists of 2 alpha subunits and 2 beta subunits, whereas myoglobin is just a single subunit that exists independently. So hemoglobin, since it consists of 4 subunits and each of them have their own heme prosthetic groups, each of those hemes can bind their own oxygen molecules, and hemoglobin can bind up to 4 molecules of oxygen. And hemoglobin is not found in muscle tissue, it is found inside of red blood cells. But the difference does not quite stop there, as we are going to see, although, as I referenced, a single alpha or beta subunit of hemoglobin, if we isolated it all on its own, it would look nearly identical to a single subunit of myoglobin. Now, here's where the other differences come in. Even though both myoglobin and hemoglobin are oxygen-carrying molecules, their dissociation curves for how they like to bind to oxygen are very, very, very different. Myoglobin shows a totally logarithmic curve. What that means, in the blue graph you can see, which is shifted to the left compared to hemoglobin's sigmoidal curve, myoglobin has a much, much higher binding affinity for oxygen. What this basically means is that it's going to be much, much, much more difficult to convince myoglobin to release its oxygen. As we interpret the curve here for myoglobin, the only way you can start to pull oxygen off of myoglobin is when the partial pressure of oxygen in the area gets extremely low, like practically towards 0. This means that the only time myoglobin in skeletal muscle is going to let go of its oxygen is when those muscle fibers are practically out of oxygen, such as when they've been exercising for a very long time. As such, myoglobin operates a lot more like an oxygen battery, in which the stores of oxygen are only depleted when there is no other oxygen left. On the other hand, hemoglobin, with its sigmoidal curve, displays what we call cooperative binding, meaning that the curve starts out looking something a little more exponential in how it starts out, and then it ends up looking a little bit more logarithmic. So ultimately, this produces a sigmoidal or S-shaped curve, indicating that at lower concentrations of oxygen, hemoglobin has a low affinity for oxygen. But at higher concentrations of oxygen, hemoglobin has a higher affinity. And this actually works out perfectly with how hemoglobin circulates throughout the body as blood flows. When hemoglobin in deoxygenated blood makes it into the pulmonary capillary bed, where oxygen concentrations initially are very low, when hemoglobin in deoxygenated blood flows into the pulmonary capillary beds where it's about to be exposed to a very high concentration of oxygen, the affinity rises, and now hemoglobin can be convinced to pick up a whole lot of oxygen to carry that oxygen back to the left side of the heart. But for the systemic capillary beds, where oxygen concentration is going to be much lower, because those tissue cells are rapidly using oxygen in the course of cellular respiration, the oxygen concentration is much lower, and the hemoglobin, which is currently tightly holding onto its oxygen, as the concentration rapidly falls, the affinity is going to drop, and hemoglobin is going to want to give up its oxygen so that we can deliver said oxygen to the cells and the tissues that need it. Now, the question is, well, why does myoglobin show one kind of binding affinity curve, and why does hemoglobin show the other? This cooperative binding that we see with hemoglobin is dependent on physical communication between the amino acids that make up the different subunits, indicating that the alpha subunits and the beta subunits have to physically be able to contact one another, and since myoglobin only consists of one subunit, it cannot do it. So myoglobin, we can conclude, would be totally inappropriate in trying to do the job that hemoglobin does in the blood. That being said, myoglobin is perfectly suited to do what it does in skeletal muscle. Okay, so at this point, when we talk about hemoglobin, we can forget about myoglobin now because we're obviously talking about the blood. When we talk about hemoglobin and the idea of this cooperativity it shows, I said that this works out perfectly because hemoglobin is able to bind oxygen tightly when it needs to in the pulmonary capillaries. That's when we pick up oxygen with gas exchange. And then it needs to be able to lose affinity in the systemic capillaries so it can let go of oxygen to deliver it to the tissues. Now, let's talk a little bit about why this works out so perfectly. It turns out there are 4 different chemical factors that are always going to be influencing hemoglobin's affinity for oxygen at any given time. And these 4 things are always working in our favor to make sure things work out the way that they are supposed to. The first 2 we can mention all at the same time, the PO2 and the PCO2. These 2 factors influence what we know as internal versus external respiration. And as we're going to see here in the next few slides, we're basically just talking about the gas exchange process in the pulmonary capillaries for external respiration and the systemic capillaries for internal respiration. However, we need to take a look at how the oxygen and carbon dioxide levels influence how hemoglobin likes to hang on to oxygen in those cases. The third chemical factor is the pH, which we've already seen before in certain cases, how pH is influenced by carbon dioxide levels, so you could really make the argument that number 3 here ties in directly to number 2. But independent of that, the pH effect is known as the Bohr effect, and the pH of the blood, as we have seen, needs to be tightly regulated so that it's always right around 7.40, and as we're going to see, the hemoglobin affinity for oxygen changes fairly drastically when the pH changes. And then finally, to introduce something we haven't really talked about before, we need to also consider the concentration of a chemical metabolite that is produced during glycolysis called 2,3-bisphosphoglycerate, or you are welcome to know it just as 2,3-BPG. Okay, let's talk about external respiration first, and for this we're going to be talking about both the PO2 and the PCO2 numbers. So in the pulmonary capillary beds where we are going to perform gas exchange between the alveoli and the blood, the situation we have is that the PO2 is relatively high because we are breathing in oxygen-rich air. The PCO2 value is relatively low because the air that we are breathing in is relatively low in carbon dioxide concentration. And the pH of this blood is actually going to be a little bit higher than 7.4 because the blood at this point is replete with carbon dioxide. So having too little carbon dioxide means the pH of the blood is going to be a little bit basic. So this external respiration occurs at the interface between the pulmonary capillary membrane and the alveolar membrane, as we've seen earlier in this chapter. Oxygen flows down its concentration gradient from the alveoli, and carbon dioxide exits from the red blood cell. And you might remember from a previous chapter, I think it was chapter 18, carbon dioxide can be carried around by red blood cells in the form of what is called carbaminohemoglobin. This involves carbon dioxide being converted into bicarbonate ions through the actions of the enzyme carbonic anhydrase, and these bicarbonate ions are able to react with arginine side chains within the hemoglobin structure, and this produces an intermediate known as carbaminohemoglobin. Now, carbaminohemoglobin actually has a drastically reduced affinity for oxygen, meaning that hemoglobin really has a choice. It can either carry around oxygen or it can carry around carbon dioxide in the form of carbaminohemoglobin. So up until this point, as deoxygenated blood is flowing from the right ventricle towards the lungs, a lot of the hemoglobin at that point is in the form of carbaminohemoglobin. It's carrying a lot of carbon dioxide rather than oxygen, and that is why it's oxygen-poor blood, right? So as this deoxygenated blood makes its way into the pulmonary capillary beds, that hemoglobin is going to be exposed to these conditions that you see here because of the contents of the alveoli. The low concentration of carbon dioxide coupled with the high concentration of oxygen is going to cause this chemical reaction here to reverse itself. Carbaminohemoglobin is going to revert to regular deoxyhemoglobin, and that is going to reproduce a bicarbonate ion, which will be reconverted to regular carbon dioxide gas by carbonic anhydrase, and that carbon dioxide will be able to diffuse into the alveoli so that we can dispose of that carbon dioxide through expiration. Now at this point, that deoxyhemoglobin that was previously carbaminohemoglobin, it is now exposed to high concentrations of oxygen, and it will now readily and happily bind to those oxygen molecules to form oxyhemoglobin. So the contents or the concentrations of these things in the immediate vicinity favor carbaminohemoglobin turning back into oxyhemoglobin so that we can dump off carbon dioxide and pick up oxygen. We're actually going to see the opposite happening with internal respiration in the systemic capillaries. Coming into this situation here, as oxygen-rich blood makes its way from the left ventricle through the arteries and arterioles and into these systemic capillary beds, the vast majority of hemoglobin is in the oxyhemoglobin form. So, at this point, the affinity that hemoglobin has for oxygen is very high, and it's going to be difficult to convince it to give that oxygen up. So in order to deliver oxygen to these cells in these tissues, we need to reduce the affinity that hemoglobin has for oxygen. How do we do that? Well, we do it by exposing this oxyhemoglobin to these conditions that you see here. The tissue capillaries maintain a low concentration of oxygen, a high concentration of carbon dioxide, and because of that high carbon dioxide concentration, the pH of the blood in the vicinity of the tissues is going to be relatively acidic compared to what we're used to. So this is going to do the opposite of what we saw before. Low concentrations of oxygen are going to reduce the affinity that hemoglobin has for the oxygen that it has, and similarly the high concentration of carbon dioxide will do the same. Hemoglobin will want to give up its oxygen, and in the course of these high concentrations of carbon dioxide, that carbon dioxide will get inside the red blood cells, be converted into bicarbonate by carbonic anhydrase enzyme, and that bicarbonate will react with arginine side chains in the hemoglobin to form carbaminohemoglobin, and that will further reduce the affinity hemoglobin has for oxygen, so more oxygen will be unloaded, which can then diffuse down its concentration gradient to get into the cells in the tissue. So at this point, the red blood cells that are now going to leave the systemic capillaries to make its way back to the right side of the heart through the vena cava, a lot of that hemoglobin is going to be in the carbaminohemoglobin form, in which it is carrying around carbon dioxide that we saw before will eventually be disposed of in the pulmonary capillary beds. So we already saw this particular slide back in chapter 18, but this is another reminder that I want to make sure that you understand because it is very important. The PO2 figure literally refers just to the concentration of oxygen that is directly dissolved within the blood plasma. And being a hydrophobic gas, oxygen is not very soluble. So the amount of oxygen that is directly dissolved in the blood plasma makes up a relatively small portion of the total oxygen that is in the blood. Most of the oxygen that is in the blood at any time is carried around by hemoglobin as oxyhemoglobin. So just wanted to make sure that you remember that. Okay, so now let's move on to the pH effect, which is called the Bohr effect. You'll recall that the set point pH of the blood plasma is 7.40. We want it to maintain it between 7.35 and 7.45, obviously meaning we don't want the blood to become too basic and we don't want it to become too acidic. So we saw before how one of the major things that the respiratory control centers in the medulla and the pons are sensitive to are changes in the blood pH. When the pH of the blood changes too much, we are instructed to alter our respiratory cycles to make sure that we get the pH back to where it's supposed to go. But why though? Why do we need to do that? Well, now that we've had a discussion on hemoglobin, we can confirm that it has to do with hemoglobin's affinity for oxygen. If you look at these three dissociation curves here, the red curve represents the dissociation affinity curve for oxygen for hemoglobin at the normal set point pH of 7.4. When the pH becomes too basic, such as 7.6, for example, when carbon dioxide concentrations are too low, you can see the affinity that hemoglobin has for oxygen starts to get higher. This might sound like a good deal, but You'll recall that getting hemoglobin's affinity down in the proper context is important so that we can deliver oxygen to tissues. Meaning that if the pH of the blood is too high, too basic, we're going to have a problem being able to deliver oxygen effectively to tissues. On the other hand, at a pH of 7.2, which is too acidic and would be consistent with carbon dioxide levels being fairly high, you can see the affinity has gone down. And delivery might be better, but it's going to be harder to get hemoglobin to pick up and hang on to oxygen in the pulmonary capillary beds at that point. So our conclusion here can be that if the pH of the blood becomes too acidic, the binding of oxygen to hemoglobin is going to be too weak, and oxygen will not be adequately picked up in the pulmonary circuit. And consequently, if the pH of the blood becomes too basic, the binding of oxygen to hemoglobin will end up being too strong. Again, that sounds like a good deal, but oxygen will not be adequately delivered in the systemic circuit because at that point, hemoglobin would rather hang on to the oxygen it has rather than give it up like it is supposed to. So we really don't want the pH of the blood to change too terribly much, but we saw before with internal and external respiration, small changes in the pH in local environments due to changing PCO2 does promote oxygen pickup in the pulmonary capillaries and oxygen delivery in the tissues. We just don't want these big pH changes to be systemic in the sense that it is a widespread phenomenon. It's okay to have little pH changes in certain places, we just don't want it to be all over the place. Okay, and finally, let's talk a little bit about the effect of this molecule we may not have ever heard of before called 2,3-bisphosphoglycerate. Before we specifically talk about it, I want to ask you an important question that you may have or may not have thought about yet. How does the body prioritize oxygen delivery to tissues with the greatest need for oxygen? Now, your immediate thought might be, based on our previous conversations, that the sympathetic division of the autonomic nervous system is going to use vasoconstriction and vasodilation strategically to make sure that we divert the blood flow to the places that need it the most, like skeletal muscle. And if you're thinking that way, that's great, and I'm really proud of you because that's kind of the way I've been training you to think. But we want to look at this a little bit more specifically. So regardless of where the blood is flowing, let's just say the blood is all flowing into skeletal muscle, how can we take extra special care of that skeletal muscle which needs its oxygen? How can we make sure it gets every single bit of oxygen that it needs, even if it's already receiving the vast majority of blood flow? So the answer is this molecule called 2,3-bisphosphoglycerate, or 2,3-BPG. This is a metabolite that is regularly produced during glycolysis. You'll recall from Chapter 3, glycolysis is the first step in cellular respiration. It is a 10-step process using 10 different chemical reactions that takes glucose and forms pyruvate molecules. And those pyruvate molecules will then be shunted into the Krebs cycle, and then eventually all the reducing equivalents produced there will be shunted into the electron transport chain and will make a whole lot of ATP, right? Well, all you need to know is that glycolysis is something that is going to happen a lot in cells that are actively producing ATP. As we're going to find out here, 2,3-BPG is actually an important regulator of hemoglobin's affinity for oxygen. As cells that are actively respiring, meaning they're producing a lot of ATP and using up a lot of oxygen, they are going to produce this 2,3-BPG molecule, and this molecule will be exported out of the cell, and it will get into the blood plasma in the immediate vicinity of those tissue cells. So this BPG molecule is able to interact with hemoglobin, and it binds to hemoglobin in a small little space in between the 4 subunits of the protein structure. It kind of looks like a little donut hole. I'll have a picture here on the next slide to show you. So the idea here is that when hemoglobin binds to this 2,3-BPG molecule, it drastically loses its affinity for oxygen. Again, that might sound on the surface like a bad thing, but remember, this hemoglobin is right in the vicinity of cells that are actively producing ATP, actively using oxygen and have a tremendous need for the oxygen that hemoglobin is carrying. So basically, this 2,3-BPG molecule is acting as a signal that is coming from these cells to tell that hemoglobin, hey, all that oxygen you're carrying, we really need that. A pretty good analogy I've come up with in the past, and this is specifically for those of you in the Tulsa area, and this is Tulsa Community College, so hopefully some of you will know what I'm talking about. You might remember the old Mexican restaurant known as Casa Bonita. It's no longer in service, but at Casa Bonita, when you sat at your table, you would have a little flag that you could raise, and when you raise the flag, your server would know that you need something like drink refills or sopapillas or more chips and salsa or something like that. So this 2,3-BPG is kind of like a flag that cells can raise to indicate to hemoglobin that, hey, we need more oxygen. We need more of that oxygen that you're carrying. We need you to give us more oxygen than you normally do. And by reducing the affinity that hemoglobin has for oxygen, we can get more oxygen delivered to these cells and to these tissues than we ordinarily do. So these 2,3-BPG levels will be highest in the immediate vicinity of cells that are showing very, very, very fast metabolism, using glycolysis a lot, producing a lot of this 2,3-BPG, and this acts as an indicator of which cells need the oxygen the most, and hemoglobin will prioritize them for getting them the most oxygen delivery. Okay, so here's that picture that I promised I'd show you. If you look at the hemoglobin structure here, you can see all 4 subunits. The 4 subunits put themselves together in such a way that there's a little hole in the middle, like I said, kind of like a little donut hole. Well, this space perfectly accommodates a molecule of 2,3-bisphosphoglycerate, and it is coordinated perfectly by some amino acid side chains by these different subunits. So when 2,3-bisphosphoglycerate binds here, it causes the protein structure of the entire hemoglobin to change in a subtle way that causes the hemoglobin to not want to bind to oxygen nearly as tightly. So if we look at the dissociation curve of pure hemoglobin with no 2,3-bisphosphoglycerate versus hemoglobin with 2,3-bisphosphoglycerate, you can actually see that without this regulation, hemoglobin starts to look a lot more like myoglobin. So really, absent any other factors like carbon dioxide and the Bohr effect, 2,3-bisphosphoglycerate is a very important regulator of making sure that hemoglobin has that sigmoidal relationship with oxygen rather than the logarithmic one that myoglobin does. So like we said, 2,3-bisphosphoglycerate levels will be the highest around actively respiring tissues, the cells that need the most oxygen like skeletal muscle. So in those areas where 2,3-bisphosphoglycerate levels are very high, the affinity for oxygen drops. And you can see that evidenced by the red curve here being shifted to the right from the black curve. However, you might be worrying, okay, well, with this 2,3-bisphosphoglycerate keeping the affinity for oxygen down, how's that going to affect hemoglobin picking oxygen back up in the lungs, in the pulmonary capillaries? Well, the good news is that by the time the blood circulates back to the lungs, that 2,3-bisphosphoglycerate should have basically completely disappeared because of enzymes chewing it up and using it for other things. So by the time the hemoglobin makes its way back to the lungs, there should be no more 2,3-bisphosphoglycerate, and hemoglobin's affinity for oxygen should be restored to the maximum so it can pick oxygen back up, and then we can do this whole process over again. All right, that's going to do it for this supplemental video. As always, I thank you for your attention, and I will see you next time. So long.